So thank you so much for having here, and very good morning to everybody. And it's really nice to be here at Flemingsbury, and it's kind of nostalgic as well because I have spent all my life as a researcher here in the Noam Labs in that floor. So it feels very happy. And before going into the presentation, I would just like to ask you some questions. Maybe you can answer this to yourself in your mind. So definitely we have like more biological data than ever before today. And when you generate a new data set, so what takes longer? Is it to produce the data itself, or is it to understand what it really means? And are we really extracting the full value from this data, what we have produced? And most of us, we might have this moment as well, like, a promising signal, a gene, or a pathway. And the question is it really important, or what we see is just a noise? So as a researcher, I worked at the intersection of biology and data, and I kept seeing these small gaps actually. What an incredible data sets. But there has been limited ways for me to explore them across the diseases. And today, in the next few minutes, I will show you how Mavatar helps researchers to move from data to insights by uncovering the hidden biology and behind it without months of manual analysis. Just to give a short introduction about Mavatar. Mavatar stands for Medical Avatar. So we are a Swedish biotech company pioneering in AI and data-driven solutions for medical research as well as in precision medicine. And we are tackling a huge healthcare problem here. Despite drug development remaining an expensive and lengthy process with an estimated cost of around $2.6 billion US, and also around 10 to 15 years to develop one drug. Yet up to 75% of the patients do not receive an optimal therapeutic benefit. And this is mainly due to the biological heterogeneity across the individuals, including variability in their genetic architecture and molecular pathways, networks, and also everybody has a different disease driving mechanism. And as you can imagine, this has a serious consequences which comes with a huge human cost as well as a huge economic cost. And the bottleneck is, it's not due to the lack of data to address these problems, but the lack of early, reliable biological insights needed to guide scientific, economic, and patient-centric decisions. And at Mavatar, we believe a healthier future starts with empowering researchers, healthcare providers, and patients to make the right treatment decisions. And that's why we are leading the personalized healthcare revolution for all diseases, from research to patient care. And there are also some of the common research challenges that we researchers face, such as relying on the known literatures, and sometimes which might be a bias and which could limit our discoveries. And there are always problems with big data integrations, technical challenges needing a lot of coding experts and statistical modelings. And also there are a lot of multifactorial causes leading to one single disease. And challenges with animal testing and availability and non-availability of post-mortem tissues for research purposes, that could be also very challenging. And researching in silos, cross-disease learning, and also visualizing and presentation of the data, that could be also sometimes really time-consuming and challenging. At Mavatar, we are trying to solve these research and healthcare challenges with our proprietary DINA technology, which is based on 20 years of research from academic universities. And today, our DINA technology is distributed via two platforms. One is Mavatar Precision and another one is Mavatar Discovery. So talking about the Mavatar Precision, it's a clinical decision support platform that builds biological twins of the patients using molecular and clinical data and helps clinicians predict treatment responses and choose the most effective personalized therapies for the patients. And there it's still in our pipeline, and the plan is to launch it in 2027. But the other one is Mavatar Discovery, and that's what we are focusing today in this talk. So Mavatar Discovery, what it is. So it's a platform which helps and empowers researchers and provides them with an instant biological landscapes for any drug target, pathways or diseases by turning the scattered scientific knowledge into clear, actionable biological insights. By this, we help researchers to rapidly map disease biology, to identify key interactions, and also to explore drug repurposing opportunities, thereby reducing weeks and months of work into few hours and significantly accelerating research, supporting faster hypothesis generation and also more informed earlier project decisions. So just to give a few words about our DINA technology, DINA stands for Deep Integrated Network Analysis. It's a strictly data-driven framework that enables cross-disease discovery using large-scale public transcriptomic datasets and supporting systemic and unbiased exploration of disease mechanism. My colleague Camilla, she will add more on it to this later. And whereas the traditional bioinformatics workflows are often slow, fragmented, and they really depend upon scarce expertise, Mavatar Discovery changes this. And as I mentioned earlier, the DINA technology, it transforms thousands of public transcriptomic datasets into curated, biologically meaningful insights that can be explored and applied immediately without any coding and no pipelines and no delays. And this helps us the opportunity to accelerate biomarkers and target discovery with high confidence molecular networks, explore disease biology in a much more interactive way, and compare disease conditions across tissues, across organs, and also in different patient groups, and validate your hypothesis faster with using reproducible and standardized data sets. And saying that our platform, it supports every step of drug development, ranging from pre-target validation, patient stratification, mechanism of action, and also drug repurposing, leading to a significant reduction in time, cost, and also earlier de-risking in your research. I think the next part, my colleague Camilla, she will take over.
Hi everyone, and thank you for coming here today. I hope you enjoyed your breakfast. So as Swetha said, we use public transcriptomics data, and just so you get a quick glimpse, as of June 2025, there has been published more than 7.2 million transcriptomes. So when you have new data, you generate it in your lab, you are kind of obligated to publish it into these repositories. So we are taking advantage of all this data that is out there. And we use our technical framework, which is an open framework that studies new areas in a kind of up-to-speed way, because data is published and then we integrate it into our framework. And we use all the available bulk RNA-seq, microarray, and also single-cell data. And our pipeline processes it and curates it real time, and we use these steps: quality control, normalization, machine learning, integration, and then we create our beautiful networks. So yeah, math is nice, but as we all know, biology matters a lot more, and understanding the disease processes and the drug responses requires the analysis of vast amounts of data, not just one small data set. And in Mavatar, we leverage on transcriptomics, or the sequencing of messenger RNA to uncover these disease-related changes in the cellular state. But also at Mavatar, we believe in the importance of shifting away from single target paradigms towards systems level modeling of interactions. And what does this mean? Well, usually when you do an RNA-seq experiment, you have your up and your down regulated genes, and you find what's changing, but not how it's connected. So not the big picture. But biological function really emerges from connected systems. Gene-to-gene correlation networks capture this really well. So in these interactions define the function, not just the expression. And the perturbations that happen propagate through the network, which helps you interpret indirect effects and identify key regulators, such as targets or biomarkers. So our DINA framework reveals coordinated biological systems and highlight key drivers and expose mechanisms that remain invisible in single gene interactions. So in our networks that you observe when you access our platform, you can see that nodes represent genes, and two nodes are connected by an edge if the two genes are consistently co-expressed across all the data sets that we analyze and integrate. So just to give you a quick example, we did a fast case study because in the pandemic, clinicians were asking, well, who would benefit from anticoagulant therapy? And there has been a lot of investigation of the use of prophylactic anticoagulants, and the findings remained inconclusive. Why? Because patient heterogeneity exists. Not all patients have the same biology. And here we can explore our networks with the drug targets at the core. So we used the delta parin target, which is SERPIN-C1, I believe, and we investigated the gene expression differences between patients and conditions. And so we searched for delta parin, and in our blood network for COVID-19 patients that included more than, I think, 1.2 thousand transcriptomes. And we found 28 genes in close proximity to the target. And we plotted our network in a heat map, which is another cool feature in our platform. And we obtained 4 major patient subgroups, which again defines a bit the patient heterogeneity that we are seeing. Patients are responding, patients are not responding. And we saw this group 1 here on the left that was enriched in high expression of platelet activation markers such as ITGA2B, SERPIN1, von Willebrand's factor, and this pattern indicates an enhanced pro-traumatic phenotype, suggesting that group 1 may most likely benefit from prophylactic delta parin. And within this network, within the close proximity, we also found thyroglobulin. This encodes a protein produced by thyroid follicular cells, and it's a precursor of TG, which is precursor of the thyroid hormones T3 and T4, blah, blah, blah. But it influences a coagulation by affecting the balance between blood clotting and the breakdown of clots. So we propose that it could be used to monitor, it has been used to monitor patients after thyroid cancer treatment. But given its clinical use, we can also say that it is a promising biomarker for anticoagulant therapy and for patient certification in COVID-19. And looking at the expression of the gene in other conditions, we also saw that it is highly expressed across multiple conditions, which highlights possible relevances in other diseases. So just to summarize, we did a network-based transcriptomic analysis with our platform, and in under 5 minutes, we got to all these conclusions because it's just a lot of data summarized in very fast insights for you. And we found a transcriptomic defined subgroup that may derive benefit from prophylactic delta parin, subgroups with low coagulation gene activity, and thyroglobulin is a novel promising biomarker candidate. And the good thing is there are no limits to the queries and insights you can extract from our platform, and we can help you unlock them fast and avoid usual research bottlenecks, such as the ones that Swetha described. And everything is data-driven, so you've probably used tools like String. In String, all those interactions have been described in literature. Here we have novel ones, maybe interactions that have not been described before. Yeah, so thank you very much for the talk, and we have an upcoming webinar on rare diseases very soon if you want to sign up and see more of the platform live in action. And you can get access to a free version of our platform using this QR code. So I encourage you to do it. It's very fast and easy to use. And if you have any questions or feedback, we would love to hear from you. Thank you.